Maybe just very quick introductions. So I'm Chris Mungall. I'm at Lawrence Berkeley National Laboratory. I'm not actually part of the HubMap consortium, but I'm kind of tangentially involved in a number of ways. I work on a number of different ontologies that are part of the OBO, Open Bio Ontologies Consortium. It includes the cell ontology and specifically the Uberon anatomy ontology, which I've kind of like played a major role in developing. And as part of that work, I've worked with people like Marianne in getting the brain section of Uberon kind of aligned with a number of different existing atlases out there. So thus far, my interests have really been, I also work on projects like the gene ontology. Thus far, I have not done a great deal of work on things like aligning anatomy ontologies to spatial coordinate systems, but it's something I'm kind of like very, very interested in, and in particular doing things like spatial reasoning over anatomy ontologies. So that's, I guess, my kind of like background and why I'm here. I don't know if you've done intros in previous sessions, but I mean, if not, then Marianne,
usually do you just a background. Yeah, sure. Marianne Martin from UCSD. I'm part of the Brain Initiative Cell Census Survey Consortium, and I can just leave it at that.
Well, if we're with introductions, I am Enrique Sapena. I am a data wrangler for the Human Cell Atlas. And I came here because I'm really interested in the Common Coordinate Framework, as we are trying to also, at some point, integrate data from spatial technologies, and we'll need metadata for that. So,
great. Sean?
Yeah, so I'm Sean Hanlon. I am at the National Cancer Institute, and I am the program director for the Human Tumor Atlas Data Coordinating Center.
Okay, Tyler? Are you there, Tyler Best? Maybe not. Maybe Ajay, you can go. Oh,
I'm Ajay Pillai. I'm a program officer at Human Genome Research Institute, and I'm part of the HubMap project.
Okay. And okay, it looks like maybe Tyler doesn't have audio, but he is chatting in the chat window. HubMap. Okay. All right, so why don't we just go ahead and get started? So that, I think the initial prompt is a good one. Currently, at the moment, how do common coordinate frameworks and anatomy ontologies currently cross-reference each other? So I'm not really aware of kind of existing efforts here. So does someone want to kind of like jump in with what they know of CCFs?
I can talk a little bit about efforts that are underway in the INCF around brain atlases to develop a brain atlas reference, essentially ontology, that separates out the different components of atlases so that each one can be properly versioned and specified. Because one of the problems we're finding in brain and other initiatives is, of course, these things aren't static. They change over time, labels get reassigned. Even the spatial location of a brain region can change as new information comes in. So we thought that it was very important that there be a common way to reference the different things that go into making an atlas. And those include, let me see, where did I have that open? The coordinate system, the labels, the delineations, and I think there was one more thing in there that can vary. Oh, I think the alignment, how you align it. So I think that's important for all of these consortia moving forward, especially for brain, where we have no shortage of ways of assigning labels to parcels of brain.
And so the output of that project is literally kind of like a set of shape maps for the labeled shape maps for the brain for different atlases, or
well, there's that, but it's also how you version all of those different components so that when somebody says I aligned to Paxinos are aligned to this, which if you go to different versions of Paxinos, there's different terminologies, the underlying labels change, you know, it's a very fluid environment. So we wanted, the ontology really covers not so much how you relate a spatial chunk to a term, which is very important, but also how you reference the entire artifact.
And is there a kind of link we can put in there at the moment? Is this kind of available within Interlex, or is it a separate thing?
Right now, it's a white paper that's under development, and I'll get permission to share the link with everybody, but it's coming from another group, so I don't want to share it otherwise. I'll pull the slides out that I think are public.
And so does anyone have any kind of like comments on this for other anatomical entities? You know, I'm not sure if people, you know, we have anyone from, say, KPMP for how this is working with kind of like kidneys, or how this is, what the plans are for the rest of you know, the rest of HubMap here. Yeah, as David said, it's maybe unfortunate we have the CCF group meeting separately at the same time just now.
Yeah, I mean, I was at a KPMP ontology meeting, and they were primarily focused on the cell naming problem.
Yeah, I mean, the one I went to was similar, as well as kind of like, you know, diseases and phenotypes, you know, as well.
And I think they were going to use Uberon effectively, or a subset, the relevant kidney subset of Uberon, and the cell ontology to do their work. I did not hear anything much about coordinates.
Yeah, I did not either at any of the kidney workshops I went to.
So I can say a few words about some of the activity in HubMap. I mean, one of our groups is building a tool that enables people who are slicing different parts of the kidney to place it within some sort of a computer representation of the kidney. So at least, you know, spatially, you can position where your tissue slice is coming from. And there have ways to normalize with the size differences in the organ. I'm not exactly sure if they have ways, well, nothing much has been done. So the only place it has been used in is kidney, which is a fairly static thing, more or less. It doesn't like flop around. We'll see how it functions for other organs.
And you know, what level of granularity is that? Does that go down to individual cells, or is this more the individual kind of like, you know, loops?
No, so it's basically, you know, so typically, so for a spatial transcriptomics assay, right, they would use about 1 millimeter cube. So that's what is being placed into the organ reference frame. So it's at that level of granularity, if I remember things correctly.
So not within the 1 millimeter cube, but placing that 1 millimeter cube within a larger spatial context.
Right. And then once you run the assay, you get more details about, you know, where in that 1 millimeter cube you have the 1 micrometer slice that you used, or the smaller slice that you used, not 1 micrometer, but whatever 20, 50 micrometer slices that you used come from. I don't think we have gone to that level of resolution, but things are rapidly evolving there. So I might be somewhat delayed in my catching up with what's happening there.
Okay, and that's presumably for the adult human, metanephros kidney.
That's right.
Yeah, because one of our other prompts here is, what are our plans for doing this kind of like cross species, and depending on what organ we're talking about, there may be more or less work done in terms of mapping in some of these other species. Obviously in the brain, there's detailed rat and mouse, macaque atlases and so on, but that may not be the case for other organs.
Yes. Another thing that came up during this whole atlasing workshop that gave rise to this working group is that the QC metrics for spatial mapping and spatial registration are generally not very well defined. So people do these alignments, but having some way of judging the quality of these alignments was something that at least the brain, I don't call them brain mapping because it's not human necessarily, but let's call it brain mapping people were interested in working on.
And when you say for alignments, is this intra and interspecies alignments, or specifically?
Well, yeah, certainly if you are trying to warp data from one species into a coordinate system of another species, that would be even more critical.
But you're saying it's even the case that even for intra, it's still
James G. brought that up, from the University of Pennsylvania. He said, people say, I aligned this, but then you don't know really how good it is, and especially in a brain where you have very precise, tiny little structures, right? How well aligned you are will very much influence. Which is why I kind of feel, even though there's some who feel that that the terminologies aren't as important as the space, if you don't have a means of referencing, I think the cat fight is starting up again, excuse me. If you don't have a means of attaching labels, then I think things will be way off. Let me go shut the door, I'll be right back.
So I assume if that is the case for the brain, for which people have been doing this kind of thing for a long time, it's going to be hold even more so for other organs as well. So it sounds like this is something the HubMap, needs to address, is kind of like, just better metrics for alignment and transformations as a whole.
Okay, crisis averted,
yeah. I was just saying, Marianne, that this probably, I would assume this also holds for other organs as well outside the brain.
So.
Something probably CCF would want to address. And, the use of the labels, will be kind of important as a kind of like, a QC metric there as well.
Absolutely.
But what is the strategy for improving QC there? Are there gold standard alignments that people have kind of like looked at and validated? That would presumably be quite a painstaking curation task.
Not that I'm aware of, at least in my domain, but I wouldn't be the one who would necessarily know. But I think it's a good question to address. I know the whole issue of spatial coordinate systems for squishy organs and things that don't really, we have enough trouble in brain, which is, has a lot of landmarks, a lot of substructure to it. I can imagine in other places it's much, much, much harder.
So moving on to one of the other prompts, what standards should we use for referencing CCF frameworks and anatomy? So it says like referencing coordinates and referencing regions of interest using ontology terms. So, obviously I would kind of like promote the use of Uberon as the labeling system for for that. Other standards, that we should be aware of here, or so it sounds like for particular organs like brains, there may be kind of emerging standards kind of coming out of INCF and other places that may be useful here.
Well, I mean, Chris, that the big problem in brain is all the different custom terminologies that have come out. And there's, in many cases, only so much you can do, because depending on the modality, how the brain was cut, what techniques they used to do it, the labels aren't going to necessarily match up. So, but a general model for how you relate nomenclatures in many cases from existing atlases to Uberon terms, I think is a really important one to have. And then the referencing of the spatial referencing. Are you referencing a point, a boundary, a line, right? All of the things that you might need to consider when you're putting a spatial definition in, because I could easily see that just because you have a set of coordinates, somebody else may give you something that's not comparable to it. So.
So is it possible to solve a cross-species question without addressing a single species question? I don't know, I'm just asking. Do you have to solve one before you get to the other?
I'm not sure exactly. I mean, what do you mean by solve here? What kind of question are we?
So if we don't have a bunch of agreed upon common coordinate frameworks for one species, can we do, or for each species, can we do cross species without solving each one of the species individually first? Or are biological questions going to direct which mappings are more relevant and which mappings we don't care about? How do people think that's going to work?
Well, I mean, ultimately, I do think which ones are useful depend on what data comes. And most of the data will be from a specific species, if that's what you're getting at. I'm not 100% sure. If you reformulate it, I'll put it in the notes.
Well, if I couldn't make 5 people understand, it's probably not an important question.
No, but I think you are getting at something here. I just may not have understood it, but others may have. So why don't you repeat it?
If I can think of a better something to say, I will ask again.
Okay. Was it how useful is a cross species ontology, or is it?
No, I mean, I understand cross species in cellular nomenclature land.
Right.
I don't understand cross species in spatial land.
No, in spatial land, I think is interesting, and that's what I thought you were getting at. I didn't want to put it in your in your mouth, but I've always felt that it is the nomenclatures that cross the species and not necessarily the spatial coordinate system. But I don't know, with some of the new things coming out of development and things, one could see fairly sophisticated approaches to actually mappings. And you see examples of this in brain, where they'll take a rodent brain and sort of warp it to what a human brain would be, to say, for example, that most of the cortex in rodents is sensory, whereas in humans it takes up virtually no space whatsoever. So I'm not an expert in this, but I think the question of cross-species spatial mapping is an interesting one.
And I'm sure this must be kind of like very organ dependent as well. And that, there's different strategies you could use. There's looking at kind of like what the functional role of that region is in the two species. But it may be, there may be kind of like cellular similarities, there may be kind of functional similarities, and so on. So I think it probably depends hugely on which organ that you're looking at.
Mm-hmm.
But yeah, I'm assuming, you know, one of the intended outputs, you know, eventual outputs of, you know, projects like HubMap would be a set of, you know, canonical coordinate systems for different organs on a per species basis, and then you know, some kind of attempt at mapping these across species, which will probably vary tremendously, you know, in terms of, you know, accuracy and different metrics that can be used depending on the organ and which species barrier you're crossing. But, you know, I assume that, you know, that would be one of the goals here, because it would be incredibly useful, obviously, to project from an experimental organism onto human to understand, you know, more, you know, looking at how particular diseases may target, you know, particular subregions of that organ. You know, it would be incredibly useful to be able to kind of like map that across to human and to get hypotheses to see if some of the same systems are involved.
Okay, put that down.
But yeah, I have no idea, you know, what kind of progress has been made so far within HubMap, within the kind of like the CCF groups, in terms of coming up with some of these systems. But this seems like something that the anatomy ontologies could help with, because within the context of things like Uberon, we have made different calls about whether we believe, you know, it's not just nomenclature alignment. We you know, in various cases, we say, okay, you know, in trivial cases like the heart, we say, okay, the mouse heart and the human heart, they're essentially, you know, obviously homologous organs, you know, and we're grouping them together for biological reasons. You know, once you get down to the finer subparts, you know, we may have kind of different classes, you know, for representing how these things are different in different species. So the anatomy ontologies could be used as some kind of, you know, baseline for evaluating some of these mappings. And it would be kind of two-way, you know, a kind of two-way street as well, because once we have these kind of coordinate systems and alignments, then we can use those to inform the ontologies as well. So one experiment we did when trying to align Uberon with some of the different atlases, in particular the Allen atlases, was because these atlases are kind of like, you know, exhaustive, mutually exclusive partitions of space, you encode, even though the ontology isn't really very good at working with coordinates per se, you can still encode kind of essentially symbolic constraints that say, you know, these two regions, you know, should have no parts in common. And then you can run an OWL reasoner, and then that will tell you, oh, but, you know, in the mouse ontology, they put this under, you know, both kind of like forebrain and midbrain, for example, when really it should be just overlapping both of these rather than parts of one of these. So by using the atlases, we were able to kind of encode some of these constraints and then use that to actually feed back to some of the existing ontologies, cases where they had modeled something, you know, maybe in a not quite ideal way, and we were able to kind of like get fixes and just learn more about how people are using different kind of like terminology and so on. So it can, yeah, the two can inform one another. So we got a question from Tyler. Assuming each organ has a foundational functional unit, should map CCFs be spatially oriented to those units? Yeah, I mean, I don't know necessarily if every organ has one foundational functional unit. I mean, what would that be for, you know, the brain, for example, you know. But I think, yeah, definitely, I think the maps should, you know, take into account these functional units where we know them.
There's also multiple kind of purposes, because a lot of the landmarks and things that one might orient to have no functional, known functional association, but yet they're still important for being able to orient yourself and communicate with each other.
Okay, we've got 5 minutes to break. We address some of the questions that we went through here. Or?
Is it useful for us to think about what experimental data is gathered by who to construct common coordinate frameworks?
Should that be under use case queries for CCF or a new topic? Let's see. I'll put it a new topic. What data is gathered?
So Marianne, I mean, I guess the brain is probably the most advanced in generating or defining CCFs.
Yes, I would say so.
So is most of it from MRI images, or are there molecular things?
No, I mean, certainly the first Allen one, right, was not really based on, in the mouse, was not based on MRI. It was based on whole brain sectioning, and the stereotaxic coordinate systems are based on skull morphology. So they have these different purposes. Once MRI became available, they started to create MRI-based atlases of oxome space for mouse and rat, and of course the famous ones for human. But they come from a bunch of different modalities, depending on what you're trying to do. And of course, with the brain, the ones that have gotten a lot of traction are in experimental animals are the stereotaxic ones, because then people can inject dyes and do surgeries and what have you.
So the stereotaxic ones are not mappable easily to MRI images?
They are, but it's not as straightforward as you would think, because once you take a brain out, it often distorts. When the animal itself, how do you align it? To do that, there have been some in-skull high resolution images of mouse and rat brain. But the brain, there are algorithms for this, obviously. And the thing is a lot of the molecular type atlases that have the fine, or histological atlases that have the fine parcellations of brain structures, because MRI only gets you, right, so many, you have to section the brain. And then you have to rewarp it and reconstruct it. And some of the most famous atlases really don't necessarily even, do that. They just apply their anatomical knowledge to the histological section and do as best they can to sort of align that to the stereological, stereotaxic coordinate space. So I would say brain probably has more algorithms and more experience doing this than any other organ. But because of distortions and things, and the necessity to be able to destructively slice and re-image, it's been surprisingly hard. And some of this is coming out of the big brain initiatives, I think, in Europe, to merge all modalities into one space. But this is not something that I'm an expert in, so I might not be up on the state of the field.
Pushing on this a little bit more, so when you say that because they are not static, or I forget the word, the flexibility in the structures, now are these because they're anatomically different, or are they because they're functionally different, or or both, that the mapping becomes tricky?
So if we're talking about having a kind of a multimodal atlas where you take, for example, an MRI, and then you histologically section the brain, it's the same brain. It's just a matter of doing the computational warping to try to get them back into the same space, but trying, that's a fairly compute intensive, and also it's a compute intensive activity. If you're trying to relate it to living animals and things, then right, the brain in a living animal is not exactly the same as the brain in a fixed animal. The brain shrinks, so you have to be able to correct for that. You have to align the animal in the MRI scanner, and usually that's flat and linear, whereas in the behaving animal, right, you have various flexures and things. So in that sense, again, these are all doable things, and our atlases have certainly gotten better, but they do encounter challenges that you might not think happens. In terms of the staticnicity I was talking about in the beginning of these atlases, I mean, as we get more data, the definition of what motor cortex is changing. And also, if you functionally define motor cortex, and then you even have something like Brodmann's, sometimes they overlap well. Brodmann's, which is a histological stain, sometimes they overlap well, but sometimes they don't overlap at all when people do the careful experiments. So mapping functional, especially in brain, which is a huge network, onto discrete chunks of tissue has always been a problem. And even mapping, fMRI into structural MRI has its own, right, its own subfield because here it's non-invasive. You have the same brains, but you have to get the landmark problem. You have to, do a lot of work to get them together. But you are correct that the brain people have been doing this for a long time.
Okay, so it looks like we're about to go back. How long is the break? And then we reconvene in the same group?
Yes, I think it's 15 minutes, did they say?
I think it's 15 minutes, yes.
Yeah. Are we on break? I guess so.
Okay.
Chris, we need you to declare we are on break, so then we can go.
You're on break, yes. I will see. I may have to miss part of the next session. I don't know if it's better to miss the beginning or the end. Probably the end.
I have to get on another call at 12 my time, so. I mean, we could just keep going till 12 and then you could have a break and I'll leave.
Should we do that? Should we move on to the next topic here?
Is that okay with everybody, or did everybody want a break?
We can continue.
Okay, thanks. Sorry about this. So how might we questions? Is that what you want to move on to?
Yes.
Okay.
So wait, so if neither Marianne nor Chris are going to be necessarily around for the next session, should we just directly go to the blue session? At least we'll have one blue thing done.
I think that's what we're proposing here, just going directly to part 2.
Yeah.
Okay. All right.
Problem we think we can solve. So I think that the work that's going on in brain, which is essentially the ontology of how you describe an atlas, I think that is entirely doable and probably useful to this consortium as well, because it's not, again, the nitty-gritty of the individual structures. It's just making sure that the different components that go into an atlas can be referenced appropriately, so that if there's differences from one atlas to the next, one atlas version to the next, you can clearly specify what those are in a machine readable way. Does that seem like something that would be useful to this consortium?
Mm-hmm.
Okay. So that one, I think we could work on by collaborating.
Okay, does someone want to try and get that down?
Let's see, what did we call it? I'll just take the name from that. Where's the white paper? Anatomical Atlas Reference Ontology model. And that we can just say build on work that's going on in INCF for brain and see its applicability to other organs.
This is a recommendation.
Are there references one can include, Marianne, so that other groups know
Yeah,
what it is?
I think that let's just reference the slide set because that one is in the public domain. I don't think the white paper is being written by a graduate student and I don't have permission, even though I would just as soon share it with everybody. But I'll put that in. Where is it? I think the anatomical, the anatomical, how about something like standardized means of capturing different components. Atlases, e.g. coordinate system, reference data, terminology, and delineations. So in a machine readable form. There, I think that does it. Okay. And this one would perhaps be one leverage work. Oh, there it is. Right. See image below. And
so Marianne, is this an atlas in terms of cells or is it an atlas in terms of space?
It's basically any atlas. The problem that this was designed to solve was every time a new version of a brain atlas comes out, they change things, right? Structure names change, structure delineations change. And we're seeing this in the Brain Initiative where people who align their data to the Common Coordinate Framework version 2 versus Common Coordinate Framework version 3, the motor cortex completely changed, right? They don't overlap with each other because the definition changed as we got more data. So depending on which coordinate system you use, which terminology you use, right, which underlying reference data you use, what we're seeing is that these things are in flux over time. And therefore, for big projects which collect data over many years, they're going to have multiple versions of this. But not everything changes across atlases. Sometimes it's the same terminology, but the coordinate system changed. Sometimes the coordinate system changed, but the terminology didn't change. So for people in informatics and neuroscience, trying to figure out whether Swanson's CA3 field is the same from atlas version to atlas version turns out to be very, very difficult. And so the experts that were all brought together about brain atlasing and common coordinate frameworks just agreed that having a common descriptive ontology for the underlying artifact, this doesn't even get into, right, how you define an area. It just says you're going to, all atlases do something, right, to define areas. And so we just want to specify those. So I think it's a pretty high level, it's a high level artifact, but still very useful so that as projects evolve, it's clear where the data came from, so to speak. Does that make sense?
So kind of using ontologies as a migration tool, let's say? Kind of?
Yes, I think so.
That's essentially a data model for being able to represent all of the aspects of any individual atlas.
Yes,
So
exactly.
standardized
That's
way.
why I think it's doable because, right, it's just getting into fair practices for individual atlases so that we're not left in the dark about what the heck is going on underneath them.
Mm-hmm. And so just staying within the brain, how does this relate to, say, the ALAN have their own kind of JSON schemas for representing at least kind of some aspects of these atlases? Is that something that, we could just in terms of using the brain as a kind of exemplar here, could we maybe assume you're maybe working with them already to kind of like map that JSON representation to your standard one? Does that make sense?
That's what it should be.
Mm.
But it's in many cases, they haven't even differentiated between, some of these components. And so I think we're going to have to look into it a little bit further. But this at least gets people to think about it, I think, more carefully than generally people do, because they assume, again, that these things will stay the same over time. But as more data comes in, our whole understanding of the underlying structure is going to change in many cases. And that may not matter for the space unless all of a sudden they recalibrate their coordinate system, which does happen, right? It does happen over time, but it does very much matter for this mapping of labels onto this coordinate system.
So how much work has been done in terms of, so you've got the kind of like, is this like a proposed data model, or can I like now download like Swanson, Paxnos, or even like the ALAN atlases using this model? Or
no, you won't be. So it's right now, it's a proposed model. I think it needs to be, we're testing it across these different ones to see how well it works. So it's nascent. And that's why I think it's probably good to get input here as to whether this will only work for brain or does it also work for other organs. But at a minimum, I figure on this, even if a complete computational artifact doesn't come out, if those developing atlases and coordinate systems and CCFs start to be careful about how they document and reference these things, we'll be, even if it's just in free text, we'll still be better off than we are right now. Yeah.
Presumably you've got more of a retrospective problem with the brain, because there's all of these atlases that have existed with all kinds of different representation schemas before, but I think we've got the option to be more prospective with other organs where this group, the hub map will be coordinating a lot of the initial efforts in building atlases.
Exactly.
So,
There we go.
So, yes, there's a quick question. How easily do the researchers adapt to this standard, at least in brain, with their experience? Do you usually have trouble validating against this? Or, sorry, I don't think I'm making myself clear.
What do the researchers do?
So I'm asking about more metadata validation, as in like working for the human cell atlas. We've seen we're trying to impose a metadata schema and like try to create a standard. So one of the things that we've seen is that it's not easy to tell people you have to put the data this way or this other way, because there are different points of view. How easily?
So I don't know that. So brain is requiring people to state we're going to use these standards and we're going to put them in this archive. And the archives, as I understand this, are the ones who are going to drive those requirements. And they're required to do that if they get brain funding. So part of it is taken care of, which is you will do this. Our own experience, though, is that compliance with it it's all over the place, right? Because until you have really good tools, you don't necessarily get good data. But people are required in the cell census to map to the CCF. I know in the European Human Brain Project, the spatial mapping is done by the curation team. So people submit their brain slices and things, and it's the curation team that maps them to a common coordinate system. So I think there are different models for how that works. And that's one of the reasons that we didn't, for this one that I was talking about a moment ago, all the researcher would have to do is say, this is done according, this is the version of the atlas I used. That's all they're required to do. They don't have to do anything else. But this one is for the atlas makers. And as Chris says, it might be the biggest problem in brain. It may not be a problem anywhere else. So this is just an exploratory group.
Yeah. Okay, thank you.
But we have a lot of other things that we talked about, Chris. There are some problems that you wanted to address with cross species, and then just how do you represent spatial regions inside of ontologies, I think. Because I think HubMap does have a model for that, don't they?
I'm not sure.
But I think that would be worth investigating, because I know, and again, I'm not the person really to ask about this, but in a brain region, you could store a central coordinate, right? Because usually people will accept that the centroid in the caudate nucleus is the caudate nucleus. But of course, as you move out, depending on the technique and all the other things, people don't agree on the boundaries. But what is the best way, if any, right? Inside of an ontology, so that if I took Uberon, it would have spatial definitions. That to me is a real challenge. Seems like it's less of a challenge the other way around, that a coordinate system would suck in labels so that it would be locally constrained. But is there a global way of doing this so that one would know that the definitions across are equal, or is that a nightmare?
I mean, you can certainly, if you have everything fully specified using some kind of quantitative spatial system, you can reason about descriptors. Like you can reason that in all species, region A borders region B, or there's maybe an exception in one species where there's some other structure that's been kind of interspersed there. So, I think maybe that's the point that we will be aiming at in the future, when we have these fully specified descriptions. But at the moment, it seems, at least until we have that, we've got these kind of like manually curated spatial descriptions, some of which are encoded as ontology axioms. So we have, as well as the part-of relations, for example, which we have in Uberon, kind of things like adjacency relations as well, where we can say region A is adjacent to region B. And you can even have those kind of constrained in a species-specific way, which we've done in a few cases by essentially deriving these from the atlases. So,
so would that be a worthwhile little pilot project to do with the information that's coming?
Yeah.
Because that sounds actually very neat.
So this would be a separate. Should we copy and paste this blue box if we think we have a separate?
Yes.
Yep. Okay. So. See if this works.
And that's the one that I think is the most interesting of them, because that would make the ontologies very powerful.
So have people investigated all the geographic information system data models? I know that, you know, they, at least if they're just sticking with the Earth, they have a single coordinate system to map to. That eases up a lot of things. But a lot of issues like, you know, knowing vegetation cover and how you take one vegetation cover map and overlay that as time changes with another vegetation cover map, ignoring the coordinate system or ignoring the Earth, for example. I'm sure those are important questions they have addressed. Maybe there are ways we can think of adopting some of what they have done for standardizing such relationships.
I put that in the notes up above, AJ, too.
I guess most of the time there's not really a transformation problem in that you'll always have a geolocation, which is, you know, the Earth doesn't kind of work in the same way.
Right. No, I understand.
Not all the time, but sometimes. In California.
Yeah.
But I just put a note that I do think that would be one of the first places to look, because they're obviously associating labels with and have spatial descriptors. And I think that there are fairly good spatial algebras and things, right, Chris, that people have looked into for ontology. The problems that we've always had in trying to do that, at least in brain, is that these are kind of complex three-dimensional structures. And so they might like have one point up here that touches, but the rest of them are not adjacent to it. So, you know, how do you kind of capture the nuances of what's going on? But I think it's worth, I think it would be a really powerful artifact if one could take these coordinate systems across species and be able to ask these types of questions without having to recalculate them from every atlas.
So Chris, I vaguely remember you guys had some spatial ontology. Is that of relevance, or am I dreaming of something?
No, it is here. Yeah, so it's got a number of kind of like, this is the spatial relation, BSPO. So it's got spatial relations as well as kind of like just ways of describing different kinds of compartments. So that would give you the vocabulary for being able to describe these symbolic kind of ontological relations between things. So, you know, as well as saying A is adjacent to B, you can say kind of like, you know, A is in the anterior part or the posterior part of B. And, you know, the BSPO kind of gives you kind of like a species neutral way of kind of like using some of these, because there's, you know, there's various different kinds of anatomical axes that you could use depending on the organism. You know, there's things like kind of anterior, posterior. These get a little bit, you know, challenging in kind of regions like the brain as well as I recall. But, you know, for other body parts, you know, I think it, you know, there's, you know, it should be more straightforward to kind of like describe things like, you know, an anterior posterior axis or a, you know, just kind of superficial versus deep axes like that.
Oops. I'm sorry. I'm really sorry. I have to pop off, but if there's anything else in the second part that you need me to do, just let me know. But thank you. Bye, everyone.
Thanks, Mara. So I'll just put a link in the SPL here. And so there's, as well as BSPO, there's just ARO, which is the oboe relations ontology.
Yeah, I think a lot of these are there already in the document that Marianne linked, the presentation.
Okay, good. Because there are some spatial relations that maybe would not apply to the brain that would apply to other structures as well. So, well, I guess they apply everywhere. But if your focus is less on the kind of like this, the neural tissue, once you've got the kind of like the the venous system and so on, then you've got kind of like relationships like tributary of and kind of like branches from, things like that.